Hello students, so welcome back to the channel. In this video, we are going to study about Calvin cycle. Now see, in the previous videos we have studied that light reaction is the one which is performed in the presence of light in grana to prepare ATP and NADPH. So question is where these ATP and NADPH will be used. So they are used in the synthesis of food, which is processes occurring in the stroma. Okay, it is occurring in the stroma and it is done with the help of an enzyme Rubisco. So this process is not directly depend on light, but it is using the product of the light reaction. And it is observed that immediately once the light become unavailable, even then also this process is continuing. After some time, it will stop. And if light is made available again, this process will start. So some people says this is dark reaction also, but dark reaction is a misnomer. It is not dark reaction. It can occur in the light also, but it is independent of light. Is it clear? So this reaction is dark reaction calling is a misnomer. We should not say it's a dark reaction. And they want to know how the reaction is moving. So just after World War II, several efforts were put by the different scientists, and Melvin Calvin is the one who has used radioactive carbon, that is C14, and he used algae to study which is the first carbon dioxide fixation product. And he found that the first carbon dioxide fixation product was of 3-carbon organic acid, which was also known as 3-PGA. Later on, he discovered the complete biosynthetic pathway. We call that pathway as C3 pathway. Why? Because the first stable product is of 3-carbon. We call it as Calvin cycle also because he is the one who has discovered it. Now, this is the enzyme which is used in the reaction. The carbon dioxide is accepted by the acceptor, that is RuBP, which is of 5-carbon, and that 5-carbon is your ketose sugar. It is ketose sugar. So this is the one enzyme which is going to catalyze the reaction, which is the most abundant protein. And this is earlier, its name was carboxydismutase. And this enzyme is called Rubisco. Why? Because it is having carboxylase activity and it is having oxygenase activity also. It is having affinity for both. But binding is competitive. Okay, binding is competitive. Though Rubisco is having great affinity for carbon dioxide when carbon dioxide and oxygen are equal. But in nature, they are never equal. So it is the concentration of oxygen and carbon dioxide which will determine who will bind. Right now, we are discussing only its carboxylase activity. Now see the steps of the Calvin cycle. First step is carboxylation, and that is, it is catalyzed by the enzyme that is known as Rubisco. Clear? And here what happened? Carbon dioxide will combine with 5-carbon compound, that is RuBP. And right now we studied this enzyme is having affinity for both oxygen and carbon dioxide. So carboxylation is the most crucial step of Calvin cycle. Again, it's a statement question. Please learn for your competitive. It's the most crucial step of Calvin cycle, where carbon dioxide is doing the carboxylation of RuBP. And one carbon dioxide molecule with one RuBP will make 2 molecules of 3-carbon compound. So it results in the formation of 2 molecules of 3-PGA. Now this is acid. Now this acid is having more oxygen, or you can say it is having less hydrogen as compared to the carbohydrate. The amount of hydrogen in relation to oxygen is reduced. In sugar and other carbohydrate, the ratio of hydrogen to oxygen is 2:1. Here we are finding hydrogen is reduced because it is PGA. So we need to now do the reduction. So this step is the one in which hydrogen has to be added to glycerol 3-phosphate to produce carbohydrate. And we will use both reduced NADP as well as ATP. And finally, if you see, reduction phase will make triose phosphate means sugar, and which is used to prepare by different pathways. It is used to prepare finally glucose. So 6 turns of cycle are required for the formation of one molecule of glucose. Now we have to regenerate RuBP. First carbon dioxide acceptor of Calvin cycle is RuBP. So regeneration is using, again, it is very important because regeneration of RuBP is crucial if you want the cycle to continue. And this requires, again, ATP for phosphorylation. Now, let us see how many ATP are used per carbon dioxide and how many ATP and NADPH are used to prepare one glucose. See, in carboxylation, no ATP and NADPH is used. 0. Neither ATP is used, neither NADPH is used. In reduction, 2 ATP and 2 NADPH is used per carbon dioxide. Is it clear? For one carbon dioxide, 2 ATP and 2 NADPH is used. During regeneration, only 1 ATP is used, but no NADPH is used. We have to perform 6 Kelvin cycle because this is a data of 1 Kelvin cycle. So for 6 Kelvin cycle, then only one glucose molecule will be made. So 6 carbon dioxide will enter, 18 ATP will be used, and 12 NADPH will be used. Then one glucose is prepared. So I think so this numerical area is also might be comfortable for you. To make one glucose, 6 turns of Kelvin cycle are needed. Thanks for watching. I hope this video might be helpful for you. In the next video, we are going to study about C2 cycle, that is photorespiration.